Hello, everyone. This is Rafi, and I'm here with my friend Peter of Hyperlipid, the very famous or infamous blog, depending on which side of the diet spectrum you live. So welcome, Peter.
Hi, Rafi. It's great to talk to you. Okay.
Yeah, this is the first time we've actually spoken with video, audio, all of that. We've only emailed or chatted in the comment section of your blog, actually, for quite a few years now.
Yeah, on and off a little bit. It's been. I feel you're a friend rather than just someone I see out on. That's for sure. I hope that's okay.
Oh, absolutely. I'll start by saying that so the reason why I actually went into science, or a very big reason, was trying to understand your blog posts. I was struggling to really grasp the concepts in their entirety, and I was spending so much time on it whilst I was a customer service agent for TomTom in Amsterdam. So I'd have one window open, answering client emails and customer emails, all of that. And the other one would be the electron transport chain and your blog, and then a picture of it and trying to make sense of it. And I was spending so much time that I just said, well, I might as well get a degree for this.
Right. Okay. Here's the question then. It's probably you've already. The question is, do you think it holds water? Do you think I can be wrong?
Oh, you can definitely be wrong, but it definitely holds water, because I think as we've progressed over the years, I would say it's grown. I would say that the evidence base for it has grown stronger. Because you do address the paradoxes. In fact, in one of your latest blogs, I think it was on looking at DHA and comparing it to linoleic acid in terms of the fattening potential. And that was one of the paradoxes that you're trying to resolve. And I think that was a key sort of personality trait of the blog, if you will, where you weren't shying away from them as we see happening all the time in medicine or in research, but rather you were looking for them, seeking them. So that was, I think, very formative for me as a young researcher.
The DHA is easy. That does explain itself quite well. What doesn't is the alpha linolenic acid. That really should be bad.
Right.
But that doesn't come over that way in the current publications. So this is something I'm well aware of that I can't ignore. It's something that I'll pick at until I get something that makes sense. Oh, Raphael, right. I do apologize. Excuse me a moment. I just have to do a quick thing with my phone. Sorry.
Sure. Please go ahead. And so for those who don't know, alpha linoleic acid is the plant version of omega-3 and DHA and EPA being the animal forms that are active, biologically active, if you will.
But it should be mitochondrially targeted. It shouldn't target the peroxisomes. So why doesn't it behave like super linoleic acid? It should be like linoleic acid, but more so. And you really, really have to look at the fine detail of the studies to try and work out whether that still holds water. So that's at the back of my mind. I wouldn't say I'm working on it, but at the back of my mind, I'm very, very aware that puts a great big hole in my ideas, which is fine.
Right.
That's what
Okay. Yeah. And so in the blog post where you were addressing it most recently, you were talking about where beta oxidation can happen. And it can happen in the peroxisomes, or we have the mitochondrial metabolism of those fatty acids. So maybe you want to give just a brief overview of why we even have peroxisomes, as far as we know, and why they are shuttled in one area or the other.
That comes back to David Speyer. Dave Speyer from Amsterdam, I think. And he's a kind of Dutch version of Nick Lane. So, but he's looking at the evolution of LECA rather than LUCA. LECA, last eukaryote common ancestor. So when the alpha proteobacterium ended up inside the archaeon, and that was the birth of the eukaryote line, all sorts of hell broke loose at that point. And Dave Speyer was looking at the generation of reactive oxygen species during the mitochondrial oxidation of very long, very saturated fatty acids. And he and I think independently, realized that if you had a lot of flavin-derived electrons being put into the electron transport chain, then that drove reactive oxygen species generation. And he was looking, and still does look, primarily at avoiding the toxicity of having too long and too saturated a fatty acid. So palmitic is fine, stearic is fine. Much above there, you really don't want to put it into a mitochondrion. And when you get up to C26 or C28 or C30, these have very, very high flavin inputs compared to their NADH inputs. And so his idea, his concept was that the peroxisome was an evolutionarily beneficial development that allowed the shunting of very long chain saturated fatty acids primarily away from the mitochondria, so that they couldn't generate the reactive oxygen species by reverse electron transport through complex 1. That was his basic idea. And it was very, very nice of him, because I've kind of started thinking along these lines, and he emailed me and said, you do realize that this and this and this. And again, he's primarily looking at avoiding the toxicities of very, very long chain saturated fatty acids. And he feels the evolutionary solution was the peroxisome, which still generates reactive oxygen species, but not next to the mitochondrial DNA. And clearly in the amounts that tell the cell something. So the peroxisomes still produce peroxide, hydrogen peroxide, which is why they're called peroxisomes, and it still is a signaling molecule. And the level of research into the signaling based around peroxisomes is just at the beginning. People have just started to realize that the peroxisomes do signal, that they signal using reactive oxygen species, but there really aren't any models. The isolated mitochondrial preps are there, and they've been there for ages, and quite what they tell you is anybody's guess, but they're there. But the isolated peroxisomes have just started to be looked at, and there are now one or two reviews out for looking over the last few years, and people are starting to ask questions. But how they signal and what they signal is going to be a little while before it comes. Ultimately, it'll signal, it'll be reactive oxygen species. It will signal insulin resistance, and because there's too much very, very long chain fatty acid coming in, I reckon it will be another source of the reactive oxygen species that are designed to limit caloric ingress into the cell. And it will end up that way. Whether low levels of peroxisomal signaling will facilitate insulin signaling, I think that's much more of a moot point. But high level production of peroxide is going to shut down insulin signaling. That would be my guess. And that kind of, there are suspicions in the early data coming out. But as I say, people have just started to produce controllable models of peroxisomal activation, where they can put a control step into whether peroxisomes are produced or not produced. So they've then got intact cells that they can turn on and off peroxisome in signaling. But they're just so happy to have a model when you read the reviews. But they've not actually found out what the model says yet. But it'll come. And obviously, it does come back to there is an evolutionary drive for the very long chain fatty acids to go to the peroxisomes, and obviously DHA and EPA, well, particularly DHA ends up there. Of course, there's another paradox, because there's no need for that to happen, because they're full of double bonds. You would have thought you don't drive the reactive oxygen species production if there are double bonds in there. So why are DHA and EPA primarily targeted to the peroxisome when they could theoretically be targeted to the mitochondria? But then if they were targeted to the mitochondria, you might end up getting very, very fat if you've got a super version of, of linoleic acid can make you fat with 2 double bonds. What's DHA with, has it got 5 or 6 double bonds? I lose track. So that, there may be some evolutionary advantage that getting fat's good for you when winter's coming on, but getting too fat might slow you down when the mammoth is going to stomp on you. So
right. And the first thing that comes to my mind is the different caloric percentage in which we'd have encountered linoleic acid and more monounsaturated and saturated fatty acids. And is that, you think, maybe reflective of the peroxisomes and the mitochondria in that sense? Is it reflecting how many calories of these we're likely to encounter and thus handling them differently?
My feeling was really they were probably signals, as in surrogates, for the availability of animal fat derived calories in small amounts. Sorry, we have...
Oh, hello.
Oh, that cat. Oh. Oh, dear.
I love it. One of my favorite podcasts, they always have their cats in the background walking in front of the camera. I actually love it because I really love cats. It always makes me laugh.
I never thought we'd end up with 2 fluffy cats. This is the least fluffy of the fluffy cats.
Really?
But, oh yeah. There's one of them is just so fluffy you can't really tell there's a cat there, and she looks massive. But you weigh her and she's 5.3 kilos, which for a oh come on cat, you can't do that again. No, she's gone around the back of the screen. This,
of course.
But no, I was because there was the work by Sauer, Sauer, and he found that the both particularly EPA but DHA as well are coupled to signaled through G protein coupled receptors, which obviously are very, very ancient. And that to me said that they are primarily signaling molecules. What are they saying? Well, they're saying you've either caught some fish, which seems unlikely on an early human basis, later perhaps, or that there's a decent amount of animal fat around because the DHA comes in with the animal fat. Maybe that's a signal that small amounts of DHA say you're going to get a large amount of palmitic and oleic acids coming through as soon as you finish cooking. Yeah. That was one idea. I haven't really followed it through, and I'm not sure how you could kind of prove that, but it seemed just logical to me. Yeah.
And
I enjoyed
so
it. Really good. Yeah.
Right. I'm thinking back to during my master's degree where I was looking at the NAD to NADH ratio in controlled breast cells and cancerous breast cells, the MCF7 kinds, and I had to try to. There's this compartmentalization that's happening in the cell, and where you measure the redox is very important because you're not going to get the same values. And this was quite tricky. This was very, a lot of trial and error. Yeah, and what you were mentioning there about the peroxisome and the mitochondrial preparations reminded me of that. And that's maybe one reason why people haven't looked into it, because it's difficult, because it's hard to model it properly.
And of course, if the peroxisome is going to produce NADH, which is its primary electron product, it's going to stick it in the cytoplasm. So again, you're looking at cytoplasmic NADH, NAD plus ratios, which, as you say, can be completely diametrically opposite almost to the mitochondrial one. Again, what the body looks at on a signaling basis on that front is interesting, to say the least, which is again why I really don't like the fish oils, because if you're going to raise the cytoplasmic NADH level, they're what's going to do it. So that's almost, is it as bad as glycolysis, which is again going to raise the NADH level? And again, well, the presumption is that on balance, all of these things will sort themselves out. But once you start putting pathology in there, the way we do with modern diets, then I would be very cautious with large amounts of fish oil as a source of calories.
And so what do you make of the fish oil trials? The numerous ones that have come out over the years, and there's been a lot of dud results, but there have been apparently some positive results. I haven't looked at them too closely, to be honest, so I'm not even sure if there's ever been an all-cause mortality benefit shown in a controlled trial. Do you know?
No, I've not chased that. I've just looked at the observational stuff where fish oil intake it was prostate cancer was the core one, and at the time that was it's observational, so it doesn't mean a huge amount. But at the time, I was very, very pro fish oil. Obviously, over the years, then your ideas change. But back in 2006, 7, 8, that sort of time, I was probably supplementing 5-6 grams of fish oil per day, and I forget why I stopped doing it. I don't think anything particularly went wrong or anything like that. I probably ran out and didn't replace it.
Yeah, as often happens with supplements.
Yeah, I couldn't see that there was any benefit.
Right.
What I was trying to do on an athletic basis or semi-athletic basis, I could do with them and I could do without them. So it was just another, why are you taking a supplement? You should basically be eating a proper diet, and you shouldn't really need the supplements. Yeah.
Right. Yeah, and I think the one of the trials that I skimmed was talking about 5 grams of. I think I think it was total omega-3s, 5 grams. I can't honestly remember if it was for heart disease or neurodegenerative disease, but that's what they were pointing to as the relevant dosing.
Yeah.
So it's not a lot calorie-wise, but it's potentially a lot signaling.
But whether you can have a proper signal with the wrong background, I don't know. But again, I've looked at them, and they've all an awful lot of the fish oil supplemented trials were basically inconclusive or mildly beneficial. And when you look at the research bias in their favor at the moment, it must be very, very hard to get a trial to fail, but they weren't getting massive successes. So, you look at things in their context. I mean, obviously, any given paper, you read the methods first, read the results second, and then make your own decisions. Maybe sometimes you'd read the results first and then find out why the hell the results are so weird by going back and reading the methods. But yeah, the fish oil trials, I really, when I lost enthusiasm for fish oil, I lost a lot of enthusiasm for trying to go through the trials and see exactly what they've done and whether why it worked, because ultimately, when you're not expecting it to work anymore, it's very hard to get very enthusiastic about spending 2 weeks going through one paper, which when you get a good paper, that particularly when you get one that you don't understand and is very convincing, there are odd papers like that you just have to spend time. And people would occasionally put them up on the blog and say, "What do you think of this?" And you'd kind of look at it and think, "I don't know what to think of that."
Right.
And sit there, and maybe 2 weeks later, another blog post will come out. But this is really, I think, why I don't do Twitter. Obviously, I lurk on Twitter, and I much appreciate that you do a bit like Tucker does. You let people know when there's new content with you. But I often don't have an answer. People point something really strange out, and you go, "That's really interesting," but how can that be so? And then again, you're looking in the finer points of the methods.
Yeah. I think people don't realize because there are a lot of people who claim to read a ton of papers, and I'm sure they spend a lot of time, but I don't think they I think what they do is they allot an amount of time to most papers. They allot the same amount of time to most papers. And I think that's more about feeling like they've done a lot of reading. And that's very different from doing a lot of understanding. Because there are some papers you'll read in half an hour, and there are others you might spend like 2 weeks, like you just said, on it. And I don't think that's something most people will conceive of who are doing a lot of armchair research like we do.
Yeah, yeah. And that I couldn't for to save my life write a review. If you had to go back over all of the papers from the last 20 years or 20 months, if it's a more recent review, and just slog through all of them. Certain things come out of certain papers that convince me or reinforce my biases. And once I've had my biases reinforced, I will view things in those terms. And no problem with being biased. I think we're all biased. But it definitely affects my worldview. And again, the basic ideas hopefully I've got are mostly logically self-consistent. I think to me that's very, very important. And when you get something that trashes your hypothesis, that's the paper to try and understand. And I'll spend quite a bit of time on those.
Yeah, and especially there's a way in which you can prove yourself wrong. It's at least possible if the research were done where you could point to it. And I think that's again, that's just something that's very rare. I really don't see it that much, and never explicitly. Even when it's done, it's never explicit, which is such a pity, because I think it would encourage people to imitate that. Yeah, it's a pity I don't see that in papers more often. They don't. There's often not even even the language. There's none of this null hypothesis language that you might encounter when you're learning the philosophy of science or the history of science. And in modern papers, you never see those.
The other thing which I don't think everybody realizes is that you'll read a study and they fed the mice on this, and this. No, they didn't. They did a bloody pilot study first, and they tweaked it this way, and they tweaked it that way. Nobody just pulls numbers out of the air and says, yeah, we'll spend our next 3 years researching this. They want to be damn sure it's going to work before they do it. And you tweak your model till you know it works, then you'll spend the funding on it. So it's not a matter of, oh, we just decided to feed it this. No, people sat around in seminar rooms, kicking the ideas around and kicking the pilot study results around and saying, well, if we try this, then, 5 mice, you can, I don't know whether you need a home office license just to do a pilot study, we presume you do, but there'll be pilot studies there, and it very rarely gets mentioned in a given paper. But you know that they've not just come up with these numbers for this fatty acid or that fatty acid or this amount of protein or that amount of protein. It's not just a blind guess. It's they've seriously thought about this, and there will have been a pilot study.
Right. Yeah, it's all we're lacking the denominator when trying to assess the success of that line of research, and it's something that came up in my conversation with a young PhD MD, Nick Norwitz. He's 25. He already has his PhD and is from Oxford, and is now starting his MD at Harvard, and is totally bought into the keto diet because it helped him fix his ulcerative colitis, something that I also have, unfortunately. And yeah, it was just interesting to talk to him about, because we were talking about statins and the lipid hypothesis, and we were saying that however much you even want to believe the results, if we don't know the denominator because of all the negative trials that are suppressed, how could you ever trust even the supposedly best evidence, such as the Jupiter trial, for instance, those sort of things? And for me, it's a no-go once I see that. Same with antipsychotics, by the way, the field of my PhD. It's just the number of negative trials that aren't published. For me, it's I don't even know how people can keep wanting to study those drugs, like the second generation antipsychotic. It's such a waste of energy.
Yeah. And even in anesthesia, you found that because about 15 years ago, there was a rash of anti-nausea drugs came through, and they're really good. if I was going to have a GA, I'd want someone dancatory in recovery. That would be great. But they always compared their new anti-nausea drug, was always the gold standard, was metoclopramide. And we all know metoclopramide doesn't work. It's rubbish, basically. So you hadn't got to prove very much to get your anti-nausea drug licensed. And I presume that the antipsychotics are similar.
Yeah, they have lots of adverse events, a lot of metabolic events that aren't necessarily even measured. My PhD is I'm looking at the metabolic side effects of those drugs. But that's generally not what's done. They tend to just look at the behavioral outcomes, which makes sense, but not when you look at them in isolation. And Chris Palmer, which I don't know, do you know Chris Palmer, the Harvard MD? He's a Harvard MD, and he has incredible case reports of especially one woman who went on the ketogenic diet after more than 50 years of being schizophrenic, and within a week.
I've read that case report.
Yeah,
yes. I didn't
know
who it was. No,
right.
that was quite in the early days. That's quite an old case report now. Yeah,
absolutely. And what's interesting is that I'm not sure if it's in this one or another case report, but the people could go off it and on it, and you could see the same result every time, which I think adds a lot of strength, evidence strength to these case reports.
Well, the same with the ketogenic diet and migraines. Again,
right.
The ketogenic diet is by no means a panacea for migraine, but there are enough case reports where, when it does work for migraines, the migraines kick in and kick out with ketogenic diet versus non-ketogenic diet. It's just a pity it doesn't always work.
Right. Yeah.
Of the time.
And I think these sort of instances where when it works, it's so undeniable, it's kind of victim of its own success in a way, because on the one hand, when it doesn't work, well, it just doesn't work. And yet when it works, it seems to work so well. I think it's difficult for the medical profession to know how to, what to do with that, basically. It's difficult for them to handle it.
Yeah, you have to feel sorry for them a little bit.
Right.
All of these things, it's not a panacea. It's a very powerful tool. But I'm well aware that the ketogenic diet doesn't fix everything. When it works, it appears to be absolutely amazing. But I don't expect it to be 100% effective for all weird neurological problems, that's for sure.
Right.
A lot of people, and it clearly needs giving a try. I'd never tell anybody at the start that this is going to work, because it doesn't always.
Right. I always tell them it can't hurt you, pretty much.
Yeah.
That's pretty much what I say. It's not expensive, it's safe, and there's a chance that it works. If you're diabetic, I'm very, very bullish on it.
I think metabolic syndrome is probably the easiest medical problem to get at once you have a sensible approach. But the weirder neurological problems that and some of the epilepsies as well. When who's the girl in North America, the hiking girl?
Oh yeah, keto hiking. What's her name? I always speak with her on Twitter. Her name is lost on me now, but she's had incredible history.
Her remission from the epilepsy is just amazing. It must be so galling if you're an epileptic with a different trigger, different metabolic problem, and you want to try it and it doesn't work for you. However, trying it, you've got nothing to lose, as you say, by trying it.
No. It's still there's probably some degree of protection that's offered by it, I would assume.
Yes. I mean, basically, if you're going to throw a seizure, your metabolic demand for oxygen and substrate way outstrips anything you can provide. And if you're going to have a relative hypoxia episode, being in ketosis is going to keep your nerve cells alive when as opposed to running on glucose, in which case you just can fall to pieces. So no, I can see, that would be worth having, but unless you get some sort of, to do that while you were still throwing seizures would probably be beneficial, but you don't have that feedback.
Exactly.
If you do a ketogenic diet and it completely controls your seizures, and you go out and have a slice of cake and you seizure all night that night, well, fine, that's feedback. Don't do it again. But using it as a metabolic protectant, which I would agree, I think is very, very likely to happen, when it doesn't stop the seizures, would be hard to maintain the enthusiasm. Whereas, I say when I started with ketogenic eating, I didn't really have any reason to do it. I just met a friend who'd done it for weight loss, and he said it's interesting metabolically. So, and that was Atkins induction, and I just read Atkins' book, and I just discovered the internet and PubMed, and I found the whole thing just fascinating and believable. I'm not sure that it's quite as right now as I thought it was in those days. I think there's more subtleties to it. But all sorts of small weird things happened that made me stick with it. Particularly, I lived at the osteopath. I had low back pain. You carry the cross of ulcerative colitis, that's bad. I carry the cross of low back pain. It's not nearly as bad because you can always take ibuprofen and feel a bit better. But I used to go to the osteopath on a regular basis, and I suddenly found that I could sit in the car to back out of my drive, to back out the drive slightly to the left, and then back in someone else's drive to the right, and then spin the car out and drive backwards. We lived on a cul-de-sac, dead end road, and I suddenly found I could turn around to look at the back of the car without in pain. I went in a few months later to my osteopath. I was driving past and stopped the car and got out and said, "Is Andrew around?" And I said, "Sorry, I'm back to see you, but my backache's gone." He said, yeah. He said, I've met a few people that have told me that, where they've either gone gluten-free or they've gone low carbohydrate and their back pain's gone away, but he never told me that. but there was precedent there for him, and he felt that he clearly knew there were issues going on. But then, and he seemed a very genuine person. I don't think that he was keeping the info away to keep his revenue stream going. He seemed a nice chap. I think it just was a bit too off the wall for him to have kind of put two and two together and actually make a specific recommendation to people, particularly if you're an osteopath treating low back pain and you're going to make a recommendation against all of the cardiological dietary advice that you can imagine. And that would be moderately difficult to do.
That'll be much harder to justify if anything ever goes wrong by chance or otherwise.
Well, yes, and at the time that I went into ketosis for Atkins induction, the newspapers were full of how it was going to kill you and heart disease and saturated fat. My older son was stacking shelves in Tesco's at the supermarket as a holiday job before he went off to university, and he'd bring back cuttings from the newspaper about how crazy what I was doing was, and it I know it probably took about 10 years for him to go to near ketogenic eating, but he's there now.
Oh, cool. Oh, that's great. Yeah, I've got my parents are, I would say, borderline low carb ketogenic most of the time now. So my dad really had to, he had the hardest time switching over from his, it was, I would say in the last 6-7 years, his diet has been excellent compared to the average population, but you'd still have his dried fruit and a little tiny slice of toast with breakfast and that sort of stuff. And, still have this chronic rhinitis and things going on. And once he he suffered this this head trauma, this bilateral subdural
Yeah.
hematoma, which you very kindly helped me think through, you know, the different options.
I should have asked you
Yeah.
how it was getting on. Yeah.
Yeah, he's fine. So I'll jump to the conclusion. He's fine now, but right after it, he developed Meniere's syndrome.
Vestibular.
Yeah, and I think it's related. I think that's my guess. But it seems to be waning now after a couple of months of vertigo and what's it called? When you have the ringing in the ears, tinnitus. Yeah. So he's getting better with that, and slowly seems to be back to his old self. But yeah, it's one of these things where he really dove deep into the ketogenic diet as soon as that happened, because of course when you think you're going to die, or when you think you're not going to be, you know, capable of doing what you did before, well, that's a kick in the butt that will get you to make those changes. And it's been great for him. It's been great. You know, it's one of those things where we even gave him ketone supplements to really push his ketones up as well, and that seems to help him quite a bit. My mother is much more resilient. She doesn't feel a difference when you give her ketones, but her hunger is much more controlled than my father's. Whilst my father will have a tendency to snack, and it's interesting to see that he'll see a difference when we give him, you know, a drink of ketones. Then he'll feel like, oh, I've got lots of energy. And yeah.
That's interesting. Yeah, it's because I've never supplemented ketones, but the kind of the basic research suggests that they clearly have very profound effects on their own, whether you control the carbohydrate content of the diet or not. I've never really wanted to go that route, but I think if I was involved in head trauma, then I would consider it. Yes.
Yeah, I never offered it to them before that. It was once he had this hematoma that I said, okay, let's make sure we have some on hand, even if you were just to fall down, for example, and then he'd be, you know, just as a preventative measure, and just to encourage him to also, because it really does have an effect on appetite. So I told him, whenever you feel like snacking, I much prefer you have that than you snack on some dried figs. So, you know, that was my my assessment of it.
Yes, it's funny. The one that the thing that I find stops me feeling peckish if I am is coffee. Again, coffee is ketogenic. Caffeine is ketogenic without anything added to it at all. And I have to say, it doesn't. I drink it because I like the buzz from the caffeine. But I have noticed that I will forget to eat. Like I've been painting walls for days now, and if I'm doing nothing apart from pottering around on the internet, I'll go and have some cheese for lunch or something like that. But if I've actually got something to do and I've had a couple of coffees earlier on in the day, then just I go and I do forget to eat. Whereas if I've not had the coffee, I'm aware when it's lunchtime. I wouldn't say I'm hungry or anything like that, but I'm aware it's lunch. And it would be nice to have something to eat.
And talking about appetite, have you seen that paper from Ludwig? Oh, yes, you have. I think you wrote a blog post about it, where he's looking at the substrate concentration in the brain and how that correlates.
I have huge respect there. That he knows what he's talking about. To me, leptin resistance is the fact that substrate delivery trumps the leptin signal. The brain might look at all this leptin and say, we got tons of fat available, but there's no calories coming up the carotid artery.
Right.
You're not going to listen to how much fat there is when there's no calories coming in.
Right.
Obviously there's more to leptin resistance than that. But on a basic conceptual basis, if your primary problem when you eat is that you are losing calories into your adipocytes, then the supply of energy to your brain is going to be lower. than if you were absorbing food but not sequestering it into your adipocytes. But I'd never. It was just so nice to see that he'd actually done the calculations and come up with the, And again, I don't think it's not in the 2 hours post meal that matters, because 2 hours post any meal, you will be flooding everything with too many calories. That's just, you can't possibly be utilizing calories at the rate they're being absorbed if you've just eaten 1,000 calories of lunch or something like that. So that it's really what's hanging around 3, 4, 5 hours later that determines when you next go and eat. That's so I say I'm less interested in the per acute phase. What actually stops you eating. Actually, that's another different matter. But it's kind of when do you go and start thinking about having some more food, which over the days, months, hours, whatever, will make a difference. Yes.
And I just thought of the fact that because we were talking about substrate concentrations in the blood, and one of the observations that Dave Feldman has made, which you've probably heard of, I think. He's noticed that if you go to measure your triglycerides in the lab, you probably have to wait 12 hours or more if you're on a high-fat diet, because your triglycerides hang around for a longer time. So I'm wondering if, and I'm wondering if that plays into the delayed desire to start eating again on a high-fat diet.
I would buy that perfectly well, because of course it's not just chylomicrons. I mean, a chunk of it will be chylomicrons, but you're also going to be having, there will be elevated free fatty acids as well, and a proportion of those will be recycled through the liver anyway, which is where Dave Feldman comes to his
energy model, flux energy model.
Yeah, if you're releasing fatty acids, or if your lipoprotein lipase is splitting triglycerides on the cell surface, but they're not all entering the adipocytes. Some are just, and again, they will reflect the meal that you've just had and the meal before that. But yeah, so it will be triglycerides and it will be BLDLs and chylomicrons. Though it would be nice to. I haven't seen papers that have actually looked at those. The feeling I would have would be that the chylomicrons were immediate and then faded over whatever timescale the fat was going to be absorbed or used or not used. Whereas if you're going to have labeled fatty acids coming up in BLDLs, they've got to be processed through the liver. That might take a little bit longer. But I've not found papers. I've not hunted papers that looked at that. Dave Feldman knows what he's doing, so there's no point to just the same thing.
And he gets so much. Yeah, and he gets so much shit for it. It's sort of either you laugh or you cry, but I think it's just amazing. He's put so much effort into being very specific in the criteria he's defining. He has a study challenge where try to find me a study where LDL matters when trigs and HDL are in a good range, and nobody can find it, of course. And he does this consistently, and I'm really an admirer of him in that sense. And he gets so much shit for it from the lipid hypothesis junkies. It's hilarious.
Again, and you lose. And again, I'm glad Dave's doing it, and I was very interested in these things back in 2005, 2006, that kind of early part of the century, because I happened to have a cholesterol. I think when I measured it myself in the lab, I think the best reading I had was 9.6 millimoles per liter. I'm not sure that is in old money.
It's about 3.5 grams per liter, I think, because mine at 10 millimoles with 3.9 grams.
Yeah, thereabouts. It's quite high anyway. And I kind of looked at things and looked at things and looked at things, and there was nothing convincing anywhere in the lipid hypothesis. That back again, that long ago, we were still looking at the very early studies and things like Mr. Fit and things like that, and the Mr. Fit trialists, the people that weren't enrolled that were then still followed, and nothing I could find was remotely convincing. And in the end, I decided that because in my biases, saturated fat was good and sugar was bad, that high elevated HDL was a surrogate for saturated fat, and low triglycerides were a surrogate for low sugar. And whether you actually felt that these lipid particles were actually doing anything, or whether they were just surrogates for the things that really matters, which is if you're going to be healthy, don't eat sugar and do eat saturated fat.
Right.
So I'm still not convinced that the particles themselves matter. And it becomes very, very difficult to get enthusiastic, and even to the point of taking studies apart. When been there, done that, and really not that bothered. But Dave's working very hard at it, and he's producing really interesting results. I couldn't dedicate my life to the lipid hypothesis, because even if you're just pointing out that it's wrong, it's been pointed out that it's wrong so many times.
Exactly.
There's incremental, there's glacial improvement in terms of what people seem to believe. But no, Dave's a very bright guy, and I think he's doing very good stuff. I like it. But
yeah, no, it's great. It's great. He's so, he's always available and sharing information, and just very, very glad to know him. And we were talking about it, about this, where we focus our energy, because of course there's an opportunity cost to wasting your time in research and all of that. And I think the lipid hypothesis has just been a black hole of funds and energy and all of that. It's been terrible beyond what it's done in terms of the bad advice, just the opportunity cost for research.
Research money that's been wasted, that could have been more usefully spent. Yeah, no, I can see that.
Yeah. Absolutely. And so what are your next, what are the next questions you're looking to explore in the blog?
I have no idea. I never know. I never ever know. I go where the information takes me, and just occasionally a single paper will trigger a whole load of stuff.
Right.
Nick Lane's, when he started actually building bioreactors to synthesize primarily formate from hydrogen and carbon dioxide, that to me was just, and he got it, and he knows exactly what he's doing. He's basically got the origin of life sussed. That's just to me, that's amazing, because now he's looking at starting to look at purines and the various nucleic acids, but the problem is that apparently the problem is that they you really have to be working with hydrogen in the reactor at extreme pressure. It's supposed to be anaerobic, but people don't like working with high pressure hydrogen. If you want to launch a rocket, that might be a different matter. But no, again, that somebody told me years and years and years ago to read Power, Sex and Suicide if you really wanted to understand mitochondria, and that was a real eye-opener. And the follow-ons really around the origins of life were quite convincing, but he had no experimental data in those days, whereas he's got low pressure hydrogen carbon dioxide reactions going, provided you've got the correct pH gradient across your iron sulfide precipitates. So and to take that sort of basic chemistry and look at where enzymes came from, and the iron sulfides and nickel sulfides, or iron nickel combined sulfides, are absolutely central to all of the catalysis that we do. Now that there were odd little papers that he pointed out that really grabbed me, and I had no idea I was going to write a whole series of blog posts on the origin of life. hadn't occurred to me. And some of them are really annoying when people say things about metformin. Metformin is another thing that
Yeah.
you kind of, it's very interesting. It's a very dirty drug. It does many, many things, is the trouble. And what it does at therapeutic concentrations and what it does at 1,000 times therapeutic concentrations are quite different. But it's a dirty, and I kind of look at it as a blockade of the glycerophosphate shuttle. But because it's a dirty drug, it does all sorts of other things as well, and you're never really sure if you're using it as a model for what happens if you blunt insulin signaling, or what happens if you blunt the signal for insulin resistance. It's a very, very intriguing drug, but the studies are a bit messy, to say the absolute least.
Yeah.
You need the eye of faith, and that's a bias coming out again.
Right. I think there's his name is going to come to me. Schulman. I think Schulman has a great paper on metformin. Gerald Schulman.
He's done familiar. I can't picture.
Yeah, and he doesn't subscribe to the typical mechanism of action, which is complex 1 mediated, and so that was very interesting. I think he also linked gut bacteria to it, interestingly enough, but he did mention, I think, the glycerophosphate shuttle.
Yeah. Well, again, you go back to bacteria, and there's this concept that the bacteria largely control your metabolism. Yeah. So there's the fasting-induced adipose factor, where if you starve your gut bacteria, they make you liberate fat.
Yeah.
Because it's their fat. They help produce it in the gut. They help process it in the gut, and they've helped you store it. And when they're hungry, they will release the fat from your adipocytes, keep you working, so you can go and hunt and catch something and kill something, so they get fed. And again, that was a concept I found very convincing. But of course, exactly, there are glycerophosphate 3 shuttles, not shuttles, but the glycerophosphate dehydrogenase is present in bacteria. It's not just a mammalian thing. So and again, whether within a bacteria, whether you can get metformin into a bacterium at levels that will inhibit complex 1. And I think it might have been Schulman who feels there is a complex 1 blocking site on the outer surface of the mitochondria, because obviously complex 1 spans the inner mitochondrial membrane. And if you're blocking near the CoQ pocket, well, the CoQ pocket's right on the outside of the membrane. And he feels, if he's the person I'm thinking that we're talking about, that there is some degree of complex 1 blockade without having, at cytoplasmic concentrations, without having to imagine 1,000 times concentration, 1,000 times concentration within the mitochondria, which I think has been debunked so many times now that it just doesn't happen. It's very simple to work out what volume of the liver is made of mitochondria, and if there's 1,000 times the amount of metformin in the mitochondria, there should be a certain amount in the liver. But the numbers that come out are just cytoplasmic, maybe 2 or 3 times plasma concentration, because there is some concentration within the cytoplasm. But I think it gets into doesn't get into the mitochondria at all. And again, if it's Schulman, he says, well, where's the transporter? The mitochondria just don't let any stuff in or out. So anyway, there are lots of very, very interesting things. I also started looking, well, where am I going next? Yes, you asked.
Yeah.
There's the paper. I've written the post. I might not have posted it yet. It's sitting there waiting because I got caught up with COVID-y type stuff and things, and lots of other stuff going on. But it's been long accepted that plants and algae produce superoxide extracellularly, and that when you find superoxide, when you find not super hydrogen peroxide, when you find hydrogen peroxide in seawater, which you do in an algal bloom, it's the algae that are producing it. And then they started finding hydrogen peroxide in the bottom of ocean trenches. So then it's not coming from photosynthesis. There's any light down at all. And the idea has been floated that bacteria might be using hydrogen peroxide, extracellular hydrogen peroxide. They secrete it from the surface using one of the NADPH oxidases. And it's a signaling molecule for the kind of energetic state of their cells. And again, just like insulin signaling, there's a basal level that's produced when they're static, but if they're supplied with nutrients, they secrete a lot of hydrogen peroxide and do a lot of growing and dividing. But if you get the hydrogen peroxide above a certain level, then they die.
Right.
Okay. So you're now looking exactly the same with insulin signaling and peroxide as the second messenger. And there's a paper where they basically the insulin signaling system is basically upper metazoan. Yeah, so there are some metazoan clades that don't use insulin signaling, but everyone accepts that really from sponges upwards, we have insulin, we have insulin-like peptides, and we have very recognizable insulin-like receptors, and so all of the metazoans right up to and including us use insulin. Beyond that, it was no, that's it. It's a metazoan thing.
Yeah.
And then they found that plants make a cysteine-rich peptide that interacts with a plant cysteine-rich receptor that does kind of pretty much what insulin does. And they've got nothing in common with insulin and nothing in common with the insulin receptor. But if you take that insulin molecule and drop it onto an adipocyte from a rat, it will promote glucose uptake and fatty acid synthesis and fat storage. So these are utterly non-related. And the amino acid sequences have got nothing in common with each other. And they then went on to look at yeasts. There are peptides from yeasts that again have nothing in common with insulin whatsoever, but they sure as hell drop onto adipocyte insulin receptors, and they found it from something very similar in a flagellum, a ciliate. I forget which one they use now, but so these things are there, and they floated the idea that there's convergent evolution. There is a basic underlying reactive oxygen species signaling cascade, which the metazoans have developed insulin to work on. The plants have developed something very, very much like insulin to work on, and so have yeasts, and so have ciliates. So underneath it all is the reactive oxygen species generation, which is going to be the utterly core basic signaling system. And that's being used by bacteria in the ocean to signal growth. And you feed them, they make hydrogen peroxide, they grow. If you feed them and you throw catalase at them, they won't grow.
They make water. Oh, they don't grow. Do they make water?
No, they don't grow. No, they have to have okay. I'm talking about catalase or superoxide dismutase.
I think it's catalase. I think so.
If you scavenge free radicals or you stop them producing reactive oxygen species, they won't grow. You can feed them as much as you like. So the signal actually probably goes back to bacteria. And that's if I hadn't got sidetracked with building work, builders, flooring, painting, and I wasn't looking at the COVID numbers, because as we sink into the mire in the UK, then that I would have published that. I probably ought to go back and have a redo. But there is a little lead there that the reactive oxygen signaling system is utterly basic. And I think there are, even if you have bacteria which have got electron transport chains, then that's the source of the reactive oxygen species that are being used for signaling. Why they put them extracellularly is an interesting thought. I have no idea why they do what they do. But then the mitochondria put a lot of their reactive oxygen species outside the mitochondrion, nucleus. So, and to talk to the insulin receptor and to talk to many other things. So no, that's kind of, I'm kind of interested in that at the moment. But
it's fascinating.
Yeah. And Nick Lane has updated his website as well. I did it Thursday at work last week, for odd practical reasons. They needed a vet. I normally work 4 hours, well, 9 till 2, 5 hours, but they needed a vet all day for practical, basic. Everyone's self-isolating for COVID now. There's no one to do the work. It's not quite that bad, but they needed me, and I said, yeah, I don't mind doing a full day for you. But there's a gap in the middle, and I'd eaten my lump of cheese I'd taken in with me. So I sit on the internet and in my lunch break, and Nick Lane's updated his site, and he hasn't updated his website for 2 years now, something like that. And suddenly there's all these papers that he's published. So there's going to be a lot of origin of life reading going on. And I think he might have a new book coming. I thought, I can't find it again now. But when I first Googled Nick Lane publications, there was an image of a book that I didn't recognize. So I don't know, I can't find it again since. But there might be a new book coming. So he's always worth reading. So.
Yeah, I started his Sex, Power, and Suicide, the first few chapters. I never finished it, and I'm ashamed to say that because I really should have. There's no excuse for it, and I'll get back to it. Someday I will. And you're right. I think it is key to understanding mitochondria and the fact that you can't escape reactive oxygen species signaling.
No, they're cool.
Yeah, their core. I think that forms a lot of the basis of my understanding of metabolism, really. And I don't even see how it could not be that, in a sense. It's
yeah. Well, it looks like if you go back to organisms that have evolved to conserve what things were like a very long time ago, then reactive oxygen species are still very core to them. I'm not saying that modern bacteria are not highly evolved. They've been evolving for longer than we have, and they have not changed the core signaling system. And I've been interested for a long time how basic is insulin signaling, and the answer is it's not, but it's an add-on to the ROS signal, which is, as far as I can work out, is as basic as it's going to get.
Yeah, and since it's so basic, let me try to ruin the podcast by bringing up COVID. Yeah. So how can we use this knowledge of reactive oxygen species to help us think about COVID, if we can? Maybe I'm making an assumption here, but something tells me that your understanding of metabolism also informs how you think about this disease.
No, well, yes and no, basically. The problem is, I think that the catastrophes that we're going through, and they're real, it just amazes me that we have a virus which targets metabolic syndrome, and
right.
It seems to target it fairly well. And I don't think many or any of us get out of life without developing metabolic syndrome. It's not a matter of if you become diabetic. The question is, how old are you when you become diabetic? And if you're 120, that's great. If you're 65, that's not so good. And I suppose people do get to very high ages without necessarily having metabolic syndrome, but I think it's quite rare, certainly in the current environment at the moment. And it's always right from the beginning, when it became obvious that it was elderly people with visceral obesity, or elderly people with fatty liver, or elderly people, younger people with poor oxidative metabolism, it's targeted something which is ubiquitous and which is fixable in weeks.
Yeah.
Not about in 2 or 3 days, but it's fixable in weeks, or sidesteppable in weeks. And again, it comes back to any of us who, if we're glucose intolerant, you can live with a glucose intolerant on low carbing, but you're not fixed. But whether that would be enough to get you through a viral infection like this is an interesting thought. I don't know. I suspect it might be. And again, you've got all, I mean, Tucker is the person, really, isn't he, to talk about physics, and because he looks at the higher level stuff that I mean, I'm interested because I felt that infusing intravenous soybean oil with somebody who was developing adult respiratory distress syndrome possibly wasn't a good idea. So that was way before COVID. If you were going to die in the intensive care unit of ARDS, then it was fairly predictable from the amount of linoleic acid present in your bloodstream at the time either you were admitted to the ITU or you were admitted to hospital. I forget which, it came from the papers. So I mean, you could almost blame the COVID deaths on the cardiologists over the last 50 years.
Yeah, I completely agree. It's sort of picked up the mat under which we tried to we tried to hide all our disease, and this has sort of ripped it off, and now we have all of this exposed, but we're being distracted by the keys that are jingling in front of us, and we're not understanding the implication and why it's hitting us so hard. And there's a sense where it's a very basic concept to understand that the state of your population changes figures like the IFR and how deadly something is. And it's one of my fears is that we're not going to learn from this, that we're not going to realize we put ourselves in a very weakened state, and that this virus would have impacted us quite differently had we been able to take some precautionary measures in terms of metabolic syndrome.
Yes, yeah, but the problem is that you're still so far away nowadays from acceptance of what drives metabolic syndrome, that it's extremely hard in the face of and for people to accept that diet has anything to do with whether you get a virus or not. That people who have gone low carb or ketogenic and suddenly found that they still get, I mean, I still get colds and flus and things like that. Very bad, and they're not very bad, and they've gone very quickly. I still get them, but compared to how things were beforehand, then I'm relatively respiratory virus immune. I don't get a lot of problems with it, and it has never crossed my mind that I've become seriously ill with COVID. Just I told my line manager at work that if I die of COVID, you can type my epitaph as he never expected to get ill. It just never crossed my mind. Once you realised primarily targeting elderly people, I think that my plan was try not to be elderly and try not to be diabetic. And to be quite honest, trying not to be elderly was A, bit pointless, and B, probably didn't matter. It was just try not to be diabetic. But this is it's a very biased view. It's just how I view life. And if you spoke to my wife, she would say, "Oh, everything's diet as far as you're concerned." Right.
I don't know if you know, Peter, but over Christmas, I caught COVID. Both my parents did. My brother, his wife. And so we all went through it. It was fine. It was a bit weird for my brother. He had a lot of aches and pains, and had some strong dips in energy, and then he'd feel fine, and then his energy would dip. And he has definitely has more visceral fat than I would say me, my mother, or my dad. I would bet a lot of money on that, and doesn't eat the same way the three of us do. And my dad's 73. Like we mentioned before, he did have a serious health issue with his head, and yet it really wasn't a big deal. I mean, it was like you wouldn't have noticed anything different, to be perfectly honest, than if it was typical flu you get maybe every 1 or 2 years. And it's amazing because so there is someone in my family who died of COVID, actually, is the husband of my cousin in Italy, in Milan. And he was a 65-year-old man with very serious heart issues. So he had metabolic syndrome, and he died, unfortunately. And his wife didn't catch it. They were in the same space, so we know that not everyone catches it in the same space. And it's just it is anecdata, but it fits perfectly with what we know, basically.
Or what we believe.
or what we believe, yes, indeed.
Because we all look at with the same biases at the same data. But no,
absolutely.
We think we might have had it as well, but the Royal College of Pathologists in the UK is restarting its COVID webinars tomorrow. No, tomorrow, Wednesday. We listened to all of them through the early part, and it became very, very obvious that some people have a marked serological response, and some people who can be quite ill, hospitalized, recover, and they have antibodies, but they're way below the level you would pick up with a commercial serology test. So we never bothered getting. My wife was in London late in February at the peak, basically. Was on the tube train. There was a cancellation on the tube train. Everyone had to get off the tube train and go onto another platform. And there were about 20,000 people all trying to get through one airspace. And 4 days later, she coughed really badly for a week, and then a bit more for 3 weeks. She was febrile for a day. The kids got tummy upsets for a week, something like that. It didn't twig at the time. It might have been COVID, but apparently that's what kids do. And I didn't get it. And we were very close airspace. And again, we never got tested because back in February, nobody was testing anyone unless you came from Italy or China. And it wasn't worth shelling out for the serology. It's £100 a shot in the UK to buy a private serology test, when you know that unless you have a very high antibody count, that you may just be being missed. You may be seropositive. It's really, really helpful if you know you're seropositive, because, well, the UK government is ramping down serology testing because they're worried that if people come up seropositive, they will stop following the rules. Yeah,
of course.
Which is fine, because did you listen to that guy, the ICU clinician from Lewisham General Hospital who worked through the first wave with no PPE at all?
No, do you know his name? It doesn't ring a bell.
I've got a link to there's a link to it on the blog, and it's a podcast. But he said at the start of the first wave, everybody in the ICU got blood sampled, and all the youngsters were negative, but he was positive. And he said, I'm seropositive. I've been exposed. I've not been ill in any way, shape, or form at all. The best thing I can do is make sure I stay exposed. And he worked the whole of the first wave in London with no PPE.
Yeah,
because he understands immunology. He's not Chris Whitty, who is our chief scientific officer, or Ballards, who's the chief other way around. Whitty's medical officer, scientific officer. He understands. The guys there working in the ICU. He understands immunology, and the concept of ramping down serotesting when we desperately need seropositive people you need to set up separate places for elective surgery and A&E. Yeah, so you separate buildings, and the routine hip replacements, knee replacements, whatever, your ticket to be a surgeon working with those people should be that you're seropositive.
Right.
Yeah? You have to have been exposed. You're not going to carry the virus. You're immune. You can deal with people who are in their 70s or 80s, and that's what you should be doing. Or failing that, then the other role for people who are, and obviously people who are seronegative should be nursing the COVID patients until they become seropositive, then they can go work wherever they like. Or you could look at it the other way around and say, well, okay, these people are seropositive, they can nurse the COVID patients. But then you're trying to protect everybody from ever catching COVID, which
it's like the wind, you can't stop it.
It became very obvious from the Royal College of Pathologists lectures that you were not going to stop this virus. It's you know, and to be honest, it's going to be really, really, really interesting to see what happens in New Zealand. That's going to be
right.
Yeah, because they've got a completely susceptible population. I forget how many of the very frail elderly Norwegians have died from the vaccine.
I think it was 29 last time I saw.
Because this is, you know, these are the people who are very, very frail, and these are people who'd stood no chance with COVID at all. But there's quite a big population in New Zealand, and what are they going to do if they start losing patients after vaccines when they've been through what they've been through in terms of lockdown? That'll be interesting, and it'll also be an extremely interesting field experiment in terms of actually how good the vaccine is. Because when are they going to vaccinate everybody? When are they going to open up? What's going to happen when they open up? How many people are going to die of COVID when they open up? Will they ever open up? Obviously, and the other really interesting one is Israel, because Israel are vaccinating people left, right, and centre, and we'll see. Until they get 2 doses in a couple of weeks after the second dose, I don't think you can't make any comment. I wouldn't expect any vaccine to work well unless it's a live vaccine, which obviously these aren't. I wouldn't expect a live vaccine to work on an inactivated vaccine to work on 1 dose. And though the RNA vaccines are a technology, I've never seen these types of vaccines ever used before, so I don't know what the response time would be, and whether you would get any protection from the first dose. I mean, they talk about such and such a percentage, but to be quite honest, with the way the studies were set up, I'm not hugely convinced that they very much. And the other one that's interesting is Public Health England looked for reinfection, whether you're immune once you, and they talk about field virus infection being on a par with 95% effective. But actually, when you go and look at the paper, I forget, 4,000 people, they don't know that all of them actually had COVID. Some of them are presumptive because of COVID. Some of them are seropositive, but quite a small proportion. And they've had 40 or something reinfections. But when you go through the fine print of those 44, I think it was, one of them is almost certainly, they're convinced it's a reinfection. The other one, maybe not. But because they got PCR positive on the other 42, doesn't mean you're infected. But that was what they were using to decide if people were reinfected. So I can't conceive of how having the field infection would be on a par with having the single gene vaccine, a single protein vaccine. So no, a lot of time for people becoming seropositive, it's your marker that you actually had the disease, and you should then get on with your life. And if you can go and help in ICU, go and help out in ICU. Some people say ICU. I was brought to, I lapse occasionally. I was brought to ITU, not ICU. And there was in the UK, intensive care was sort of hand cream. So the medic that taught me, they always talked about the ITU, but everyone else says ICU, but it's interchangeable. But no, intensive care is a form of hand cream. But,
Yeah, I think there's a paper that's been shared recently by my friend Gabor Erdosi, and the paper showed that it had 6 months of data, and according to that paper, that there were 0 cases of reinfection
Yeah, I
in
don't think
that paper. Yeah.
you can get PCR positive again. I've no
Oh
doubt
yeah,
about that.
for sure,
But
for sure.
I really don't believe it. I can't see. I saw
Mhm.
Gabor put up the comment that when he gets vaccinated, he wants it. There was a Twitter comment. He wanted it an intranasal.
I was just about to bring it up. You read my mind, so
Yeah.
please tell me what you
Oh
think.
yeah, no. But he knows what he's talking about.
Yeah.
It's use IgA surface immunity. It's quite nice because I kind of kicked that idea around not that long ago, and it's lovely to see convergent thinking that other
Yeah.
people have great respect for, are kind of thinking along the same lines. You
Mhm.
sure as hell want IgA, otherwise you're all going to cart it around. But if you don't cough or sneeze if you're vaccinated and you're asymptomatic and you don't cough and you don't sneeze, are you really going to infect anybody? No.
Right.
You're more
Yeah.
likely to if you did cough or sneeze, but I think if you've got IgA all over your cell surface,
Mhm.
cell membranes, mucous membranes of your airway, that's going to do a lot more than having a few IgGs sloshing around your bloodstream, because I ain't going to do anything in your upper airway. So no, I saw Gabor's tweet on that sort of basis, and thought, yeah, I really like that. That's good.
Yeah, I thought it was a good specific list. I liked it, and it's funny because when I got tested in France, it's less expensive in the UK, so I could afford it because I'm not even on the national health system anymore because of my vagabonding around Europe for work. But essentially, I was antigen. So I did the 15-minute rapid antigen test. I was positive as predicted. I was PCR positive as predicted, and my serology was negative, also as predicted, because I was symptomatic at that moment. And of course, I don't even need to do serology now. I probably would have antibodies. There's a good chance, although I don't know if you think I could still be negative and have my IgA do the work.
It depends how much you've been exposed. I'm guessing, okay.
Yeah.
You saw the Japanese paper where the seroconversion went up and up and up and up through the summer, because they never shut down. They had their face nappies, but apart from that, they didn't do much else. And the proportion of the population that became seropositive went up and up and up over the 12 weeks of that they studied them. And my suspicion is that if you have a one-off episode, get better, you may be seropositive, you may be below detectable levels on the particular test that's being used. But if you're infected again and again and again and again, then the chance of actually having a measurable level of antibodies goes up. So, like booster vaccines. So I do wonder whether the high seroprevalence in Japan is just that they never locked down, and they kept getting infected or exposed as it went through the population, so that you end up with a much higher seroprevalence than if you've got very low levels because people had it once and got over it. That's pure speculation. And also the Japanese got away with it because it would be a matter of deep shame to be morbidly obese in Japan.
Yes, that's true. Absolutely.
They would consider themselves a burden on their healthcare system, and it would be a matter of shame. So that would be my guess in terms of why they've gone away, unless they had SARS-1. I don't know which nation picked up SARS-1 the worst in that area. I've not really looked at that.
And from 2003, so what is that, 17 years now, all of the people who had, is it SARS-1 from 2003?
They didn't have a number, it was just called SARS. Yeah.
Right, and all of these people are still immune. I mean, they can't catch it again, as far as we know. So we have 17 years of data and at least 6 months of data in COVID. Yet it's amazing the number of people I speak to, and so people in research who have read immunology textbooks and all of that, and they all tell me, because I tell them, oh, I had COVID, so you don't need to worry about it. And I'll say it with a smile, and they're like, well, we don't know. And I'm like, really?
Yeah.
Are you sure we don't know anything? Do we know something?
I've read the textbooks as well, and I can't see anything that makes me think the textbooks are wrong. I just don't think they
Yeah.
are. It's a coronavirus. They're nasty. They can do horrible. I mean, some of the veterinary coronaviruses that we deal with are appalling, and very, very fatal, very high fatality rates. Not respiratory, they're mostly enteric. Enteric and once they move into the lymphatic system, then that's the feline coronavirus, and the cat's in real trouble then. There are precedents for coronaviruses being horrendous to have to live with, and there was theoretically the possibility that could have happened to us with this one. Something like that could have gone on, but I can't see any evidence it has.
Right. Yeah. And so, okay, I have two last questions because I know I've kept you on long enough, and this has been amazing. So the last question I'll ask you is, what are you eating? Because people love to hear that, and it'll be fun to give a quick update.
This morning I had 2 lamb burgers. Lamb burgers are my treat. Lamb mince is from my local supermarket, is a little bit more expensive than beef mince. And I had about a pound of lamb mince for breakfast, with fat added to it. That time I used butter. I normally use beef dripping. This evening's meal is half a kilo of beef mince, fried in a bit of the leftover lamb fat.
So you're an accidental carnivore.
Oh, I got caught in the carnivore trap. Did you?
Yeah,
I never said. I got caught in the carnivore trap. Basically, it was a complete lulu. I had a friend with intractable neurological problems, and I was curious whether going carnivore from keto would actually make a difference. But before I suggested this, I thought, well, I'll just try it myself for a couple of weeks and just make sure that it's practically doable.
Right.
And how expensive is it going to be, and all these things. And I did 4 weeks, and the backache that I've lived with for donkey's years was massively better on ketosis. It was just so much better. But it went another order of magnitude better when I went carnivore. So that was one thing. My gum health just improved beyond measure. It was just, and again, I've been keto, my gum health's been pretty good, but it was dramatic. There were all little niggly medical things that I'm 65 in 4 weeks' time, so I'm not a spring chicken. I don't expect to live forever, and I'm going to get niggly things as I get older. And it just went away. And then I went back and started with some leaf vegetables, I think, something, I forget what it was. Might have been cabbage, might have been Brussels sprout, cauliflower, because we used a lot of cauliflower rice, that sort of thing. And backache came back with like a, ouch. It was within 2-3 days, and I stopped the vegetables and it went away again. I've done it a couple of times since. We grow a lot of vegetables, and I do most of the gardening. My wife plans the gardening. She loves growing vegetables, and we grow really good beetroot, and I pickle it in vinegar, and I love pickled beetroot, and I can have it for 2-3 days, and then there's kind of niggly low grade. I don't really want this. So I went carnivore. I eat significant amount of dairy nowadays. When I first went carnivore, I was completely carnivore. There was no dairy. I can get away with dairy. Whether that means I'll always be able to get away with dairy, I don't know. But I went carnivore, and I'm not hugely driven by food, so I can cope with it perfectly well. I ate an awful lot of beef mince because it's dirt cheap. It's £2.50 a kilo, and it makes good burgers. As a treat, I think last night's treat was a steak. I like ribeye. and this one weighed probably half a kilo, one steak.
Nice.
Which is fantastic, but that's a treat. We can't live.
Right.
I can't live on that. We're middle class, but I would be uncomfortable spending half of the food budget just on me. It's carnivore, but basically I eat an awful lot of meat mince. I do eat offal on when I get round to it and remember it. I've got a slow cooker, so I'll throw oxtail in the slow cooker.
Oh, I love oxtail. I love oxtail.
It crumbles off the bone in the slow cooker.
Yeah.
Lamb's hearts in the slow cooker, they're pretty good.
Oof, delicious.
Yeah, all the chewy bits get really soft and they're fine.
Yeah.
If I roast them, the chewy bits go crunchy, and that's okay as well.
That's fine too, yeah.
Yeah. So, but I'm basically carnivore. A little bit of cheese, very, very occasional low carb dairy, cream, stuff like that, but not very much. Mostly cheese and meat. And that's. I got caught in the carnivore trap, and I've been there for 18 months now. Whether I will stay there, every now and again I experiment. I had a couple of roast parsnips over Christmas dinner alongside normal Christmas meal, and it was all right. But I'm quite comfortable carnivore. It's socially disastrous. It just in terms of going around to a meal with friends or anything like that. Yeah. And I've got good friends who are vegetarian. They're low carb, they live on eggs, dairy, proper vegetarian, but nothing silly. And if we're going to go around there, then and my friend's comment was, well, you can live on eggs and cheese that night, aren't you? That's fine. I can live on cheese. I'm carnivore by accident, and it's a complete. Oh, and that was the other thing, is that I did the PEG 400 test. You
did? Interesting.
Permeability before I went carnivore.
Okay.
Back when I was doing something pretty close to the optimal diet from Krasniewski, and my intestinal permeability was absolutely perfect. On the molecular weight curve, I was absolutely on the curve at the very, very bottom. I basically overlaid the normal lower limit of intestinal permeability. So I had very, very good intestinal tight junctions, yeah? But still, when I went carnivore, the low back pain went away.
Wow.
Low back pain, to me, low back pain is primarily gut related. Again, it's a very, very long time I wrote about Alan Ebringer. He's professor of immunology at Professor Emeritus of Immunology at King's College. A good friend of John Yudkin. They were buddies at the time, and he picked up that ankylosing spondylitis in the spine was tied to HLA B26, and this was the first human leukocyte antigen to be tied to a specific medical condition, and it was very tight, very convincing. And I was told this in a lecture. I happened at the time to have a good friend who had ankylosing spondylitis, radiographically diagnosed, not immunologically diagnosed. And I mentioned this to the lecturer. She went, "Oh God, I didn't know whether to say that or not. There's always somebody who knows somebody who's got ankylosing spondylitis." But Ebringer basically said that it's triggered by gut bacteria. It's triggered by Klebsiella in your gut. And if you feed the Klebsiella on starch, it will make a protein and enzyme to debranch the starches, and that looks to the immune system very, very similar to one of the collagen subtypes in your spine. So you make antibodies to the pullinase that the Klebsiella makes, and those antibodies lock onto your spine. They label your spine as being foreign, and you've got an autoimmune disease. Way great. And one of the comments, a girl who'd used this technique wrote a book about living on a zero starch diet, and she got Alan Ringer to write the foreword for it. She did. He's a really nice chap. And he commented that if you've got ankylosing spondylitis and you're HLA-B27 positive, this is very, very, very much, you should be doing this. Basically, it'll get you out of your wheelchair. But he also threw in the comment that even if you're not HLA-B27 positive, you might just try it anyway, because these monogenetic problems are not always monogenetic. There will be other triggers and other bacteria, but the one they'd got the tool for was Klebsiella and the enzyme pullulinase. So I got blood tested for HLA-B27. I'm negative. I was so disappointed. I met Ebringer once, and we were chatting about backache and things like that. And he said, well, I can tell if you're HLA-B27 positive or not by how you get out of bed in the morning. How do you get out of bed? Do you get out or do you roll out? I said, well, I roll out. He said, oh, you'll be positive. If you can't just get up and stand up, then if you kind of got to roll yourself over and very carefully get your socks on, then... And I was negative, so I clearly had some other trigger factor. Despite having perfect, on test, perfect gut integrity, I still got significant improvement on the back pain score by going from slightly grubby keto, highly saturated fat-based, through to a full carnivore. And that to me said, oh, geez. And again, you think that the primary defense system on the plant is to damage a gut. It's the bit that physically comes in contact with. So yeah, no. So that's I fell into the carnivore trap. If I can get out of it, I probably will do. I'm not here on a moralistic crusade or anything like that. It's a pragmatic thing. And I don't mind. I can't see me backing way off on meat or anything like that. But if I found that I can eat this, this, and this, and it's not carnivore, and I do fine, I'm not worried. I don't have to stay carnivore. But at the moment, it's dead easy, and I do feel extremely well.
No, that's fantastic to hear. And I had no idea you'd done the PEG 400 test, and it's fascinating because. So I fell into the carnivore trap as well, because I was going to the carnivore conference that Amber O'Hearn had organized in 2018. And I thought, well, if I'm going to go, I should do a little, a few weeks of it before the conference to say that I can speak about it in first person. And I did it, and my ulcerative colitis improved. And so I still have it. It's not fixed. I still get, I estimate about 50, 60 days where I have blood in my stools during the year. And pretty much every day there's some diarrhea, although there's also normal stool. So I would say I'm a mild to maybe moderate case. But what it's done for me is that, first of all, there's sorry for being so graphic, but no indigested plant food in your stools, which is very uncomfortable.
And I can believe that.
Yeah, and it's impossible for me to get bloated now. It's not possible. I can eat and drink as much water with my meal as I can. I'm never bloated. Whilst before, if I had large meals, I could feel like this was a bit too much for me. So this amazed me because it showed me that you can genuinely force feed me. I will not be uncomfortable if it's only meat and even dairy, actually, funnily enough. So it's improved things for me. I would say a good 30-40%, but I've got quite a ways to go yet. And so the fact that you mentioned that it seems like intestinal permeability might not be able to explain the improvement in your symptoms, that sort of triggers my bias to think, well, given the diet and lifestyle I have,
I think the test is as good as we've got. It's not perfect. It's a lab test. It's a lab
Right.
test. It's going
Fair
to be
enough.
correct 19 and a half times out of 20 or 19 times
Right.
out of 20.
Yeah.
It's going to have 5%.
But
failure.
it does reinforce my bias that given my lifestyle is I mean, look, I go to sleep at 10, I wake up at 6, I do it consistently, I take cold showers, I do, heat exposure, I work out 6 days a week. I mean, my lifestyle is fantastic. I'm very lucky to be able to do it. So I'm assuming, and probably I shouldn't, that my intestinal permeability is going to be as good as it can be given all these efforts I'm making. And yet this autoimmune disease hasn't resolved. So it sort of speaks to me that there's other triggers. There's something else than the gut permeability.
It's not just starch. It's not just because
Yeah.
Crohn's is HLA-B27 associated as well on a sort of basis. But no, there are other triggers.
Yeah.
And the work we've got is only so.
Yeah.
It's not perfect. Best we got, but can't complain
Right.
about it. And if you get a lab work that says your intestinal permeability is awful, and you change your diet and it improves, that's worth knowing.
Yeah.
But to me, I had symptom improvement when I didn't see how I could possibly have had laboratory numbers change. So, it did bring home to me that, yeah, the peg testing is nice, but it actually response to intervention it's a bit like celiac disease testing. Then I wouldn't tell anybody or suggest anybody got celiac disease tested. You just do the elimination diet, do it completely.
Yep.
That will tell you an awful lot more than whether your anti-entomysic or antibody count is high, low, or in between. Yeah.
And I'm going to be exploring for my ulcerative colitis. There are 3 things that I'm considering. I have to go into a deeper level of ketosis for an extended period of time, because in the way I'm eating, I'm between 0.4 to 1.5 BHB in my blood, which is pretty normal level of ketosis, but not quite therapeutic. You could argue. I think the higher levels might be more therapeutic. So I think I really need to do that for a good 2 months, maybe, to see if it's going to make a difference. And then the 2 other interventions, because I've been reading a lot of the clinical trials on UC, because I wrote an article for the paleodiet.com, and basically it's as you can imagine, it's not a disease that's treated particularly well. But what does seem promising, and it's still early days, is a probiotic called VSL number 3, which is apparently very expensive. So I don't know if I'm going to be able to afford it. And fecal microbiota transplants.
Yes.
And and what's interesting is they were both, the side effects were very low. And I have to say I was surprised, because I was actually expecting significant side effects. Just, when you mess around with gut bacteria, you can expect something to go, but they were very low. And so this, for me, it's very important that I can try interventions safely, because I'm doing them myself. I don't have a doctor really except my Twitter doctor friends, right? And it's not the same. So those are the 3 things I'll be exploring, and hopefully one of them will work.
Well, I hope there's some good for you, Raphael. It would be nice. Ulcerative colitis is not a nice condition to have.
No.
But I can see there's a lot of logic to that. And I say, yes, fecal transplants essentially free.
Right. Yeah, I don't know if I'd have the courage to do it myself yet, but I will consider it, because there's a lot more crazier things I've done myself in the past. So, yeah,
it would be nice if you've got a correction on that. Again, it's not always from what I've read, it doesn't always work. Whereas for C. difficile, it almost always works.
Right.
But no, that'd be great. That'd be great.
Yeah. I'll report back on that. I'll report back on that.
Yes, please. Yeah.
Yeah.
Okay. Well, I'm going to go and feed my children, I think. Raphael.
Yes. I'm seeing that we're at our hard stop. So thank you so much for your time, Peter. This was fantastic. And I'm sure we're going to get a lot of comments after the podcast. We've covered a wide range of topics, so this will be fun.
Good, good. Enjoy. All the best then, Rafi. I say.
Yeah, thank you so much.
You're very welcome. It's been a great chat. Thank you very much indeed as well.
Cool.
Okay.
Thanks.